Understanding the principle of soil chemistry will make engineers better stewards of the environment. And this is topic 10. Almost everything comes from soil. The food that we eat, the clothing that you have, and the fuels that we use today. Is there a difference between soil and dirt? Dirt is displaced oil. Dirt came from soil. The father of modern soil science is Hans Jenny. He said that it is embarrassing not to be able to agree on what soil is. Soil means different things to different people. Engineers look at soil as something that is moved around by equipments that will make it suitable for construction projects like houses, roads, and other buildings. Geologists, they look at soil as a temporary phase in the global rock cycle. It originally came from rocks. It is soil now, but eventually it will become rock again. Most people think of soil as a medium for plant growth. It supports roots, it provides nutrients and water, it helps plants grow upright. There were lots of debates about soil recently. Recent NASA mission to Mars made scientists to discuss whether Mars has soil or Martian soil. This is how soil chemistry defines soil. Soils are the mixture of minerals, organic matter, and water that can support life. The question is, do you think planet Mars has soil? Soils are at the interface between the atmosphere, the lithosphere, hydrosphere, and the biosphere. Every time soils are at the interface areas, there is an incredible diversity of life. Think about the middle of the atmosphere. How much life is there? What about in the middle of the lithosphere? What about in the middle of the ocean? Amazingly, there is life. However, in the interface area, there's a lot of biodiversity. Coral reefs are at the interface between the ocean and the atmosphere. Other examples of diversity include rainforests, grasslands. Soils are at a permanent interface. No matter where you go, soils are always at the interface between the atmosphere and lithosphere, or the hydrosphere and biosphere. Naturally, we have a lot of diversity. The breakdown of rocks into smaller and smaller pieces is called weathering. Weathering can happen in different ways. One is physical breakdown. Anytime rocks crash into other rocks or fall down, small pieces will break off and contribute to soil formation. Water is a big player in weathering. Raindrops falling on rocks and other surfaces and the movement of water contribute to soil formation. Think about how the Grand Canyon was formed. The Grand Canyon was formed almost entirely from water. All the rocks that used to be in stonelands are now soil. Another contributor is temperature. Rocks expand and contract depending on the temperature, which can cause small cracks to form. If water gets in these rocks and freezes, it will expand and continue to make those cracks larger and contribute to soil formation. There are 5 factors contributing to the rate of soil formation: climate, topography, time, parent material, and biology. Differences on these factors either speed up or slow down soil formation. Is the climate the same as the weather? The average weather over at least 30 years is the climate. The climate plays the largest role in soil formation. The climate in the deserts is different, making soil formation very slow. It takes a long time for soils to form. Another contributor is topography. Soil development usually occurs more rapidly, and soils are deeper in areas with more rains. Soils are usually thinner on mountains and hills and deeper on valleys. This is usually due to the movement of water that carries soil particles down, leading them into the valleys. Another contributor to weathering is time. Time makes a factor in soil formation. Going from rock to fully formed soil can take thousands or even millions of years. This leads to an important point about taking good care for our soils. If we make soils disappear or damage our soils in any way, we cannot fix this problem in our lifetime. Soil is not a renewable resource, so we have to take care of the soils that we have. Another contributor to weathering is parent material. The parent material is the rock the soil originally forms from. These can be rocks like sandstones, limestones, marine sediments, or even volcanic rocks. These rocks broken down into smaller pieces, forming the soil. If you take a closer look at the soil, you see living things like roots, insects, fungi, bacteria, and so many other things that contribute to soil formation because they break rocks and soils into smaller and smaller pieces. Imagine if you reach down and you pick up a handful of soil. Do you really know what are you holding? Do you really know what is in it? Do you really know what is it composed of? Many just think of soil as a solid material and crushed up rocks that are really small. In reality, that solid or mineral portion accounts for less than half of that handful of soil. In general, That handful of soil is 25% water, 45% mineral, 25% air, and 5% of organic matter. The solid fraction of the soil has different particle sizes. The sand has a different particle size. The silt, the clay. These particles are defined by their different sizes. For sand, it is about 2 millimeters to 0.05 millimeter. Silt is from 0.05 millimeter to 0.002 millimeter. Clay will be less than 2 millimeter. To put these particles into their perspective, clay particles are so small that 50 million of the biggest particles of clay can fit into a single grain of sand. The clay particle is compared to the Earth moon. The moon's diameter is about 2,000 miles. The grain of sand is compared to the sun. The sun's diameter is about 860,000 miles. If we compare the moon to the sun, as if the sun can contain or can fit 64 million Earth moons. The texture triangle is used to classify soil based on the different ratios of sand, silt, and clay. There is no one best component. In fact, anytime that you have soil, they consist of too much sand, silt, or clay. We tend to have problems. These problems can be on nutrients, water, or something else. The best soils have a good mix of all three, and they are called loams. Soil structure is how the soils arrange themselves. Particles stick together and form aggregates. The single grain is no aggregate at all. Each individual soil grain are not glued together in any way. The granular structure is a small grouping of soil particles, while the blocky structure is usually larger with bigger cracks in between the aggregates. For the prismatic structure, it forms column-like structures in the soil, while the platy structure forms aggregates almost like plates stacked up on each other. The platy structures are very bad about letting water infiltrate into the soil and about allowing roots to really penetrate and grow down. Aggregates can form in different ways. They are primarily formed by living organisms like soil fungi, bacteria, earthworms, insects, and plant roots. They group soil particles together. Aggregates are very important for soils because they can help prevent erosion. They make nutrients more accessible and allow water to infiltrate to the soil more easily and make water more available for plants. The soil profile allows us to see the different layers of the soil. Each layer of the soil actually has a name, and this name is called horizon. The first horizon is the O horizon, where organic matter will be found. The second is the A horizon. This is the topsoil. The third one is the B horizon. This is where the subsoil will be found. And the fourth is the C horizon. This is where the parent material is found. And the last one is the R horizon, known as the bedrock. The O horizon, where most of the organic matter is found, is actually considered not soil yet. In fact, the best way to think about it is organic matter. It is typically composed of decaying plant materials like leaves, roots, and sticks. It is typically characterized by a dark color. The A horizon, where most of the topsoil is found, has a darker color because of the addition of organic matter from the surface. The B horizon, where most of the subsoil is found, is also called the zone of accumulation. This is where materials like clay collect in soil profile for the movement of water downward. The C horizon, where the parent material is found, is characterized by a lighter color and an increase in the presence of rocks, and in a lot of situations is called the parent material, or what the soil originally formed from. The R horizon, known as the bedrock, would be called the real parent material. This is where most of the soil above it originally came from. There are many different types of soils. Just imagine going on a road trip anywhere here in the Philippines or across the country or within your own province, your municipality, or even from your barangays. You will notice soils having different colors that supports different plants. Soils have different qualities in general. Soil taxonomy involves soil orders from alphisols until vertisols. They all have different qualities in different areas. Knowing all these different soil orders could be useful if you have a career, let's say, in soils. In general, there is variability in soils. Soils vary spatially. Let us consider the map of our country, the Philippines. You can see that there are different regions where these soils are found, and even within a province, there are many different types of soils. At the local level, soils can be further broken down into soil series. Here in the province of Benguet alone, for instance, we have the Eudora silt loam and the Summit silty clay loam. If you are curious about the type of soils in your particular area, in your particular barangay, or in the farm where you are in, the best place to find the information is in the Philippine Bureau of Soils and Water Management. Because soils are variable and you want to do soil sampling before you go on gardening or let's say farming, a lot of times when you are standing in your yard or a field, the soil where you are standing can completely be different from the soil just a few feet away. To sample soil, we can use a soil probe. A probe is basically a pole with a hole at the end and a bucket so the soil can go into. The hole at the end is sharpened, and as you push the probe into the soil will enter the bucket and be held there. The soil that enters the bucket of the probe is called a soil core. The depth of the sample you needed depends on the nutrient you are looking for, or it also depends on the lab that will be processing the samples. Most of the time, a 6-inch sample is sufficient. However, you are sampling for a farming operation, you need a sample down to 24 inches because you will be looking for nitrogen, which is an important nutrient found deeper into the soil profile. How many samples do you need to take? This is a function of statistics because the more samples that you take, the better your samples will represent the average of the field. Usually, it is recommended that you take about 12 to 15 cores and mix these together in a bucket to make what is called a composite sample. If, for instance, you are sampling down to 24 inches, separate the samples into two different composites. You needed two buckets. One bucket is for the 0 to 6 inches sample, and the other bucket is for the 6 to 24 inches sample. Again, you have 12 to 15 cores, and you have to mix these together in each bucket. Bring these composites to the laboratory individually. You need to do this. In the 0 to 6 inch bucket, you are finding for potassium and phosphorus. These are non-mobile nutrients. While in the 6 to 24 inch bucket, you are looking for nitrogen, which is a mobile nutrient. Mobile nutrients like nitrogen are found deeper into the soil profile, while non-mobile nutrients like potassium and phosphorus are found at the 0 to 6 inch soil profile. There are three different methods of soil sampling. The first one is by zone sampling. Second is the whole field sampling, followed by the grid sampling. Always take note, there is no best method of sampling. This depends on the field being sampled and what are you looking for. Whole field sampling is best when you are looking for an average for a whole field, like when a field does not have very much soil variability. This type of sampling will take 12 to 15 cores at random to the field, usually in a W-shaped pattern. Composite the sample and bring this to the lab. Zone sampling is best if we know that there is some variability in the field, especially when one side of the field might be different from another place. There are several reasons for soil variability. It could be, number one, differences in soil series in one side of the field or another. Second is there is difference in topography. And the third one side of the field has ponding or water issues. The idea of zone sampling is that you will know if you treat these areas of the field differently. To do zone sampling is to first identify zones in the field. The zones could be as many as you want, and then each field will take 12 to 15 cores, following a W-shaped pattern, and composite them together for each zone and bring the zone samples separately to the lab. Grid sampling is the last method, which requires the most amount of time and cost for the soil analysis. Grid sampling is especially useful when you want to make a map or to do precision agriculture. To do this method, you overlay a grid on your field. Each square can represent a size of your choice. Go to each intersection in the field, take random samples of 12 to 15 cores within the radius of your choice at every point, and make a composite sample for each intersection. In the field map here in this example, we have 42 by 15, which will be equivalent to 630 cores. You can just imagine how this procedure is time-consuming and also very expensive for the laboratory analysis because of the great number of samples that we have. The benefit of grid sampling is that it allows you to make very detailed map of your field or garden. You can map the following, like let's say you wanted to consider the nutrient concentration. If we say nutrient concentration, this will be more of the components that make the soil a good fertilizer. Second, we can map pH. We can map the acidity or the alkalinity of the field. Second is salinity, and the third one is anything else that you still needed in your field. A data that you think is useful and of significance to you. This is very useful in precision agriculture where you want very accurate data to cost cut on fertilizer or nutrients, knowing almost exact amounts to be applied to the field. Take note, there's no real best method for soil sampling. You just have to pick the right one for your needs. So this is the end of soil chemistry. Thank you for listening.